On Monday, right, we properly walked through photosynthesis, right? Light reactions, dark reactions. We collected energy from light. We made some glucose. It was fantastic, right? And so the last section of chapter 8 is just kind of a little like footnote where similarly to what we saw in respiration, right? Glycolysis is not absolutely efficient, but it's what evolved, it's what works. Kind of in the same way, photosynthesis has a problem, but photosynthesis happens because it works well enough. And the problem is photorespiration, right? And so that's what we're going to talk about a bit in this last little section, right? And it's going to involve the enzyme Rubisco, right? And so if you remember from the Calvin cycle, right, we had CO2 from the atmosphere. We fixed it to this thing called RuBP using the enzyme Rubisco. So the enzyme Rubisco catalyzes the carboxylation of RuBP, right? And then that goes around and that's how we get to glucose. But we've got a problem, right? Like we've said, evolution doesn't necessarily make optimal solutions. It just makes working solutions. And if it works, it stays, right? If your species keeps making more of you, then you get to keep going, no matter how funky your system is, right? And we saw that in glycolysis where it wasn't super efficient, but it made enough energy to survive. And so there you go. Photosynthesis, kind of a similar situation. So enzyme Rubisco actually catalyzes two different reactions. It's quite inconvenient, really. So it carboxylizes RuBP, right, to make PGA that we turn into G3P that we use to make glucose and more RuBP. But it also oxidizes RuBP. What a horrible, that's terrible, right? And so oxidizing RuBP is called photorespiration. So Rubisco will add a CO2 to RuBP, or it will take a carbon off of RuBP. That's dumb, right? So photorespiration, catalyzed by Rubisco, actually releases CO2 back into the atmosphere, undoing carbon fixation. So the same enzyme that we use to do Calvin cycle undoes the Calvin cycle. That's silly. Whose idea was that? So what's happening is Rubisco has an active site that will accept either oxygen or CO2. Poor plan, right? Now we've seen or talked about this kind of a situation before. Oxygen and CO2 compete for the same active site. Do we remember? That's in chapter 6. What is that called? Remember any of the words from that slide where we talked about something like this happening? There's a term when two substrates compete for the same active site on an enzyme. What was that called? Bonus review time. They are competitive inhibitors of each other. It's competitive inhibition, right? Where two substrates are competing to lock into the same site on an enzyme, right? And that's happening here. Rubisco will accept either oxygen or CO2. If CO2 gets on there, then we do the Calvin cycle, we fix carbon. If oxygen is what gets on there, then we do oxidation and we unfix carbon, right? Under normal conditions, right, kind of normal temperate weather environment, right, around 25 C, the carboxylation rate, right, so the rate at which we're fixing CO2 using Rubisco is about 4 times higher than the rate at which we're oxidizing Rubisco. And this is one of those good enough, right, out of every 5 times this enzyme does a reaction, 4 times it's fixing carbon, 1 time it's unfixing carbon. It's not the worst, right? And 80% is not the worst. 20% of the time we're losing carbon to photorespiration, right? 20% of the time we're binding oxygen instead of CO2 and unfixing carbon. but that is only at this particular kind of temperature environment situation. Enzymes behave differently under different conditions, and plant physiology is going to kind of fight against us here because we've got this little fellow right here. So this is kind of an image or a drawing, an illustration of what the surface of a leaf has on it called stomata, this structure. And it's like a little vent, right? It's kind of, you can kind of think of it as where the plant is breathing, right? So they've got these stomata and they can open and close on the surface of the leaf, right? And so generally, usually up here in kind of normal, nice temperate conditions, the stomata open to release oxygen, right? Because it's a byproduct of photosynthesis, and to kind of breathe in, to bring in carbon dioxide that it's going to fix, right? Also, just kind of like when you breathe, it's losing a bit of water, but as long as the weather's all right, that's fine. But when temperature is really high, plants have to close their stomata so they don't lose too much water, so they don't dry out, right? Which is functionally like the plant holding its breath, right? So I don't want to dry out, close my stomata. Now I'm no longer releasing that waste oxygen, right? The plant's oxygen is waste, and it's no longer letting off, it's letting in, it's cutting off its supply of carbon dioxide, which is problematic. So we've got a buildup of O2 because photosynthesis is still happening. So we've got that O2 waste product, and we're not taking in CO2. So the concentration of substrates, right? Remember things that affect how enzymes do what they do. We've changed the concentration of substrates. Now we have too much oxygen and not enough CO2. So under these conditions, we now favor photorespiration, and we're going to start unfixing all our carbon, which is a problem. We're going to start oxidizing Ruby P, which is not what we want. So a lot of the world is fairly temperate and fine. So most plants still just kind of truck on with C3 photosynthesis, doing their thing. But plants that live in really dry places, plants that occur in deserts, plants that occur where it's very, very hot, and they're going to need to keep their stomata closed more so than other plants, have evolved a bit of a couple different workarounds. Right? And so if all of those other plants are C3 plants, right, we fix carbon and we make this little 3-carbon compound. So we're a C3 plant. We've got these other guys called C4 and CAM plants. Now, both of these, we'll walk through both, actually store their carbon as a 4-carbon molecule instead of a 3-carbon starters, which is why C4 plants are called C4 plants. C3 plants make a 3-carbon molecule with their CO2. These guys bring in CO2 and make a 4-carbon molecule, okay? Now in C4 plants, things like corn, like sugarcane, kind of these grain sort of plants that occur where it's pretty hot and dry, where they need to keep their stomata closed a lot of the time so they don't dry out, capture CO2 using PEP, this a 3-carbon molecule, right? Which means that when you put CO2 on a 3-carbon molecule, you have a 4-carbon molecule. And we store it in one cell. And then what these C4 plants are going to do is they're going to separate the Calvin cycle, right? This Rubisco reaction, and kind of push it further into the cell to kind of separate it in space, right? So we can pull in all these C4 molecules and then push it kind of deeper into the cell where we're not having to deal with this kind of buildup of oxygen, the stomata problem. We're going to push it down in here and run the Calvin cycle kind of down in the middle of the plant. So we can kind of maintain the conditions that we need to so that we don't have too much photorespiration happening, right? We can pump these internal cells full of CO2, pump all the oxygen into these outer cells, right? And kind of protect ourselves from losing that carbon. Right? So that's what C4 plants do. They kind of separate it in space within the leaf, right? Between different cells. It is a bit expensive. It costs another 12 ATP to do it this way, right? Because we're going to push things through and across different cells. But it separates that process in space and stops photorespiration from happening, right? Because if you've got a bunch of photorespiration happening, you're kind of negating photosynthesis in general, which is not great, right? So C4, option 1. Option 2, for plants that have to keep their stomata closed during the day, we see in the CAM plants. So CAM, you don't need to memorize this specifically, stands for Crassulaceae and acid pathway. The Crassulaceae are like succulents, things like that, things that are adapted to kind of hot environments. Other things like pineapples, which are bryophytes, I think. Anyway, doesn't matter. Do this kind of photosynthesis. And so what these guys do is they actually separate it in time, right? Where C4 kind of fixed their photorespiration problem by kind of separating things like in space, like we're just going to pump all the CO2 into this cell and let all the oxygen be over here. These guys are going to separate it out in time. So they're going to open their stomata at night where it's not as hot, right? And now we're going to fix all our CO2 at night while we can keep things open, right? We don't have to worry about being kind of all locked up and holding our breath, right? So we fix all this CO2 at night, store the CO2 as a C4, as a 4-carbon cell. And then during the day, when we have to kind of hold our breath, kind of close the stomata, we can then take these C4 molecules and then do C3 metabolism with them, right? Then kind of push them into the Calvin cycle, right? We've got this little C4 molecule. We take the CO2 off of it, put it into the Calvin cycle, right? And do that during the day while the stomata are closed.
So how do both the C4 and CAM plants prevent photorespiration from happening?
So the C4 plants do it by, because the trick is you just don't want, wherever you're doing the Calvin cycle, wherever you're wanting to take that CO2 and attach it to Ruby P, you don't want a lot of oxygen around. That's basically the trick we're trying to make happen. So one, they pull the CO2 in and stick it onto PEP. So it's like hidden, sure. They kind of tuck it onto this little C4 molecule, right? And then in C4 plants, they take that C4 molecule and push it down inside the cell, right? And so the oxygen's kind of building up out here, but they've separated it in space. So we've pulled this C4 molecule into this other cell, kind of lock this cell down. Now we pull that CO2 back out, away from the oxygen up here, and do the Calvin cycle, right? So we've kind of separated it in space. So we've taken our CO2, put it on this little malate molecule, whooshed it down into the middle of the cell, and then we do the Calvin cycle. CAM plants just do that in time. So at night when the stomata are open and they can release all the extra oxygen, they pull all the CO2 in and fix it, tack it onto this C4 molecule where Rubisco can't touch it. And then during the day, they close their stomata, pull the CO2 off, and do the Calvin cycle, right? So it's kind of like different ways of making sure you have a high concentration of CO2 around where you want the Calvin cycle to happen, either kind of in physical space or in time. Does that help?
Yes, it did.
Okay.
Thank you.
No worries. All right. So that is the end of chapter 8, functionally.
I got a question.
Sure.
So I should know this, but how many cycles do we have to know about? I know there's the Calvin cycle, and there was another cycle that was in here earlier on in chapter 8.
So chapter 8, so chapter 8 is photosynthesis. And in photosynthesis, we have basically 4 stages. And we'll go through this when we kind of review the whole thing. The first 3 stages are all those light reactions. So capturing light, running them through the electron transport chain, and then chemiosmosis, where we generate ATP. And then the 4th one is the Calvin cycle, where we use all this ATP and NADPH to turn CO2 into glucose. So there's 4 chunks, more or less. Though some of them are pretty short, pretty tiny. All right, and so this figure that we kind of end chapter 8 with is just showing, right, because we've walked through, right, we break this into like 1, 2, 3, 4 chunks, and we break this into citric acid cycle and glycolysis and pyruvate, blah, blah. But really, chapters 7 and 8 are all basically one large cycle, right? At the ecosystem kind of environmental level, right? Neither of these processes, respiration or photosynthesis exists without the other, right? All the products of one are the reactants of the other. The products of that one are the reactants of the other, right? Right, I go through glycolysis and the citric acid cycle. I release carbon dioxide from breaking down glucose. The plants take that carbon dioxide and fix it back into glucose, which I eat over and over, right? They produce glucose, which we break into pyruvate, which we break down to make ATP, right? And we release water, which they use, right? Everything kind of just goes in circles. It's all one big kind of united process. Everything gets used and cycled over and over and over again, right? You can even see the energy kind of going and changing, right? So the light comes in here, they make their glucose. We use the glucose and release heat as we are consuming the energy, right? So every little bit of it, right, is kind of involved in the same process. So this is kind of a nice summary. If you can kind of, if you like this diagram, if it's something you can kind of walk through as you're studying, that's not the worst thing, because it does kind of pull pretty much everything together from chapters 7 and 8. It does kind of require that you know what like all these little bits mean, but that's also important. So there's that. Yeah, I like it. It's very unified field there. All right. So review. Let's kind of walk back through in some of the time we've got left, chapter 8. See kind of how we feel about all this stuff, right? Start relatively simple. Where does photosynthesis happen? Okay, plants is a good place to start. Where in a plant does photosynthesis happen? In the leaves, in chloroplasts. There we go. Yeah. Always and only in the chloroplasts. Wherever a plant has its chloroplasts, that's where photosynthesis is happening. All right. We talked about photosynthesis in roughly 3 stages. What are those stages? We capture light energy. We make ATP, yep. Yep, we use all that ATP and NADPH to make organic molecules or glucose. All right. All right. Of those 3 stages, which ones are light dependent? 1 and 2. Which one's light independent? 3. The other one? Yeah. Yeah. Good. All right. What about our favorite, the candle mouse mint bell jar experiment? What the heck was Joseph Priestley doing? What did Joseph Priestley do? Put a candle in a bell jar and what happened? Well, Joseph Priestley did ultimately die, not as part of the candle mouse mint bell jar. The mouse died in the bell jar. Why? Because there's no oxygen. And he probably I mean, there's a lot of things going on here that I don't think Joseph Priestley was taking into account. And in the short term, we're not talking about like in the life of the mouse and the mint plant, but yeah, right. He said he's like the mint plant is restoring the air. What does that mean? Now we now know. What does that mean was happening? No. When things weren't dying, things didn't die because the mint plant did what? Yeah. It was putting oxygen into the air, right? Because this whole thing, right? There are loads. You can look in the textbook. They go through like a whole hot mess of experiments, right? But this whole thing was trying to figure out like how plants do what they do, right? They aren't eating dirt, but somehow they're getting bigger, right? And so one of the things they were doing, and Joseph Priestley kind of keyed in on this, is that they're giving off oxygen, right? As part of whatever their metabolic process is. So they're kind of doing the opposite of what we do. All right. What are photons? Yeah, those little particles, right? Light particles, little discrete packets of light energy are called photons. And what are they? Why do we care? They do. And so how are they used in photosynthesis? Broadly, we don't have to get too. What can plants do with them? Yeah, plants convert them to chemical energy. That's the thing we can't do. No matter what weird little men in caves on mountains will tell you. You can't. You cannot. All right. When I did my yoga, random side note, when I did my yoga teacher training, they went on and on about these people who like lived off of light and air, and you can't. Those people are silly and making things up. It's not how it works. All right. Anyway, photons are packets of energy. They're used in photosynthesis because plants can convert them into chemical potential energy. They are collected by pigments. What are the 3 pigments we talked about? What's the main one? Start there. So photons are the light energy. They're picked up by pigments in the chloroplasts. And we talked about some different kinds of pigments. Yep, chlorophyll A, B, and carotenoids. The main one was A, perfect. Chlorophyll A was our main photosynthetic pigment, right? Because it's the only one that can really like directly convert light energy, right? Kind of bring it in, change it into chemical energy. So we've got these other guys, chlorophyll B and all the carotenoids. What do we call them kind of collectively? What are they? Carotenoids are also antioxidants. But in terms of photosynthesis, right? So we've got our main pigment, chlorophyll A, and then we have all of these, what was that term? Not yet, not the photosystems yet, pre-photosystems. It's not like a hard word, if that helps. It's like just like a whatever word. So we've got the main pigment, and then we have like, no, that's too hard a word. Accessory pigments, there we go, yeah. Chlorophyll B and all the carotenoids are accessory pigments that do what? They complement chlorophyll A by right, because chlorophyll A has a really narrow absorption spectrum. It's really good at collecting energy and transferring energy from this very specific narrow kind of range of light energies. Chlorophyll B and the carotenoids aren't as good at it, but they're a lot less picky. So they're like less good, but can do more kinds of light. That makes sense, right? So we've got these basically 3 different kinds of pigments we've talked about. Carotenoids are, as we mentioned, all oxidants, right? This kind of extra purpose that they serve. What do antioxidants do? Do we remember? Yep. So they expand the absorption spectrum and they kind of repair DNA damage that happens from being exposed to various amounts of radiation. 4 stages of light reactions. Fair enough. Right. So primary photo event, electron charge separation, chemiosmosis, right? Electron transport chain and chemiosmosis. Yes. What happens in each of those? Primary photo event, what happens? Photons are captured by the pigment. Second stage. Right, so we've excited that electron. We pull it off. Yep, put it on an NADPH at a reaction center and send it on its way. Fabulous. 3 was the electron transport chain. What's happening there? Yeah, right, because we've got, right, so we've got light coming in, we've got photosystem 2, jazz up some electrons, they go through some carriers, right? We do it again in photosystem 1, and go through some carriers, right? So we're generating ATP and NADPH and all this fun stuff, right? And then stage 4 was chemiosmosis, where what happens? Yeah, right? Because all this time we've been moving these electrons through the membrane, we've been pushing all these protons in. Where are we? Where are we during the light reactions? Where do they happen? Skip that bit. We're in the chloroplast, specifically in the, not yet. Where are we before we're in the stroma? It's a weird word. This one's a weird word. Void membrane, right? All of these photosystems and proteins and carriers are in the thylakoid membrane. So as these electrons, right, are getting pushed through, coming in, exciting electrons or pushing electrons through the membrane, we're pumping protons inside the thylakoid membrane. And then at chemiosmosis, we push those protons back through what? Enzyme. How do the protons get back across the membrane? Using what enzyme? Same as respiration, same enzyme in both of them. ATP synthase, right, which synthesizes ATP. That's where we make all the ATP during chemiosmosis. Fab. Light reactions, check. All right. So after we do the light reactions in the thylakoid membrane, then we do the Calvin cycle, which happens where? Well, yeah, in the chloroplast, specifically in the stroma, that kind of liquidy stuff that's outside the thylakoid membrane. All right. We just kind of in the Calvin cycle into 3 phases. What are the 3 phases we talked about in the Calvin cycle? Carbon fixation, reduction, and Ruby P. Yeah, regeneration of Ruby P. So what did that mean? What do we start with, right? That's kind of what we do with these things. What are we starting and ending with in each of these? So we fix carbon. We make 2 PGAs. So we fix carbon dioxide to, what's the little starting molecule that's waiting for us in the Calvin cycle? Not yet. That big long one, we'll just call it by its short name. But I think that's it. Ruby P. Yeah, Ruby P. Yeah, it's got a big long bunch of chemical words, but Ruby P works. So Ruby P, we fix CO2, we turn it into 2 molecules of PGA, 2 little 3 carbon molecules, which is why these are called C3 plants. Yeah, because we fix carbon into a 3 carbon molecule. And then what happens? Phase 2. Absolutely. We turn those 2 PGA molecules into 2 G3P molecules. At this point, 2 different things can happen, right? So we're making all this G3P, right? We're pulling in carbon dioxide. We're making all this G3P. What are the 2 options for things that happen to G3P at this point? Right, we can leave, right? We can kind of like duck out of the circle, take 2 G3P molecules and make glucose. If we continue in the Calvin cycle to phase 3, what do we do with those G3P molecules? Yep, we do that. So G3P can either be used to make glucose, of the Calvin cycle, we make a glucose, or G3Ps and make more Ruby P with it. Absolutely. All right. And then today's stuff. Photorespiration. What is photorespiration? That is, you're in the right family of words. Yes. It's in there. It's in some of those things that you said. Photorespiration is. Yes, there we go. That's photorespiration. Yeah. So undoing carbon fixation, right? Producing oxygen instead of producing carbon dioxide instead of fixing it. There we go. Right. So the word kind of makes it right. We're doing photosynthesis. We're fixing carbon to make glucose. Photorespiration is where photosynthesis kind of gets derailed and does respiration instead, right? Because we're putting off carbon dioxide, right? We do respiration and release carbon dioxide. Photorespiration is when photosynthesis kind of goes the wrong direction and starts releasing carbon dioxide. If that's not helpful, you can just disregard that sentence. It's how I kind of remember it. Anyway, all right, so photorespiration, releasing CO2 instead of fixing it. What do C4 and CAM plants do to not let that happen? So they have to close their stomata, which is part of the problem, right? Because that's holding their breath. They don't release the oxygen and they start breaking off CO2s, right? And oxidizing things instead of doing photosynthesis, basically, right? So if you're a plant, you live in a dry place, and you have to keep your stomata closed, what do you do to stop photorespiration from happening? Two things. I'll take either. They do the Calvin cycle. That is a thing they do. So the stomata are closed. So the problem is they have to keep their stomata closed. Yeah, both of them do that. They use PEP, right, to collect CO2 into a 4-carbon molecule, which is why one of them is called C4, photosynthesis, because that first intermediate is 4-carbon, not a 3-carbon. And then C4 plants do what? So the stomata are closed, which is the problem. The problem is the stomata are closed. They take the 4-carbon molecule and they store it. They move it, right? Right? Yes. So C4 plants, in order to keep their carbon dioxide from being unfixed, tack it onto PEP and move it into kind of the deeper parts of the cell, right? They basically are kind of building up all their CO2 and keeping all their rubisco in the inside of the leaf, right? So that when this oxygen builds up while the stomata are closed, the rubisco isn't even there to get kind of distracted, right? So C4 plants are kind of separating things in space, right? So that when the oxygen builds up, it doesn't kind of mess up their photosynthesis. Versus CAM plants, which do what? Yep, so they also store, they start out the same. They take PEP and they store CO2 kind of on their little PEP molecule. But how do they prevent, right? When the stomata are closed, they're going to build up oxygen also. So how do they deal with that? That's what they're wanting to, yeah. How do they stop photorespiration from happening too much? It's going to be hot during the day. They're going to have to close their stomata. So what do they? They open their stomata at night. They fix carbon at night. So they build up a whole kind of like bank of fixed carbon so that during the day, when they close the stomata because it's hot, they've got kind of like a big store of CO2 fixed that they can then use to do photosynthesis. So they separate it. C4 plants separate carbon fixation and the Calvin cycle in space, cam plants separated in time, right? They fix carbon at night, they do the Calvin cycle during the day. All right. Which is basically where we ended chapter 8. So, yeah, you got this. You guys have totally got this. So, last questions, concerns? Yeah, Friday. I'm not giving any tests tomorrow.
I mean, we passed the test, we don't have to come Friday.
I see. Yeah, no, you can't do that. All right. So